Hello everyone and welcome to Unacademy. So today I am going to talk about the process of meiosis. Now when I define meiosis, it's a simple definition should be it is a double division that gives 4 haploid daughter cells from a diploid parent cell. Now this process of meiosis was termed by Farmer and Murray in 1905. Now this term can be split as meo. Meo means to lessen up. During this process, the number of chromosome lessens up or becomes half. That is why we call it as meiosis. Meo means lessening up. Okay. So this this process of meiosis has been further divided as meiosis I and meiosis II. That is why I told in the definition that it is a double division, because first what takes place is meiosis I and then we have the process of meiosis II occurring after it. Now what actually happens? There is a diploid cell in the beginning, means the number of chromosome is 2n in case of for diploid cell, that is what it is. But at the end of the whole process, after meiosis I and meiosis II, the number of chromosomes present in each cell becomes n. That is why we call it as haploid cell. And number of such cell is 4. 4 haploid cells are formed from one diploid cell. And this process occurs in sexually reproducing organisms. Now, mostly all cells in our body are diploid. Why do we require haploid cells at all? This is because you have learned in reproduction that a sperm and an ova fuse to form a zygote, which is the first step towards the progeny, formation of a progeny. Now let us consider an example where the number of chromosome in sperm is 2n and in case of ova is also 2n. Then the number of chromosome in the progeny, or after the fertilization in the zygote, the number of chromosome will turn out to be 4n. And if we further go further, next step, next generation, then the number of chromosome would further increase, and this would happen throughout the life. But we should know that the number of chromosome in all individuals of a particular species should be constant. That is why if the sperm and ova are going to be haploid, that is nn each, then we are going to restore the number of chromosome in the zygote to be 2n, and that is what is the stable or a standard number of chromosomes representation. This is a standard representation for the number of chromosome. So that is why we require haploid cells, so that in the progeny the number is restored to 2n by the zygote. Okay, so this is the explanation of the process. This, if we start with, we would take a simple diploid cell. This is a simple diploid cell. It at some stage of its life it will undergo meiosis. This is the process of meiosis, or you can also call it as a process of formation of gametes. Gametes are formed through this process only. So there would be a diploid cell only, which will lead to formation of gametes. After meiosis I, the number of chromosomes in that cell will lead to half. This is haploid condition. Further, in next step, that is meiosis II, which is equational. Here the reduction in number of chromosomes occurred, but here the number of chromosomes is same in the progeny as well as in the parent. So here no further reduction happens, rather equational division happens. So in a way you can consider meiosis II to be mitosis process only. Now meiosis I is further divided as prophase I, anaphase, metaphase, telophase. These are the 4 phases, naming them as prophase I, metaphase I, anaphase I and telophase I. Now prophase I, we are going to talk about it. Prophase I is a rather complex phase. It is not as simple as the prophase of mitosis. It is further divided into leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. We are going to look at each of these stages one by one. Let us consider leptotene at first. This is the stage during which the chromatin fiber are going to condense and coil, forming the chromosomes. Now this is how these are visible during the leptotene stage, and these are the duplicated centrioles. The spindle fiber formation have not yet occurred, but these are the duplicated centrioles. These have already duplicated during the S phase. Okay, so these are not the sister chromatids, these are just the chromosome. That should be clear, because one major difference between a chromosome and the sister chromatid is that sister chromatids would be attached with the help of a centromere in their center, but these are all alone. So these are the chromosomes. Now zygotene stage. Before I lead you to the zygotene stage, you should learn that what are homologous chromosome. I was telling you in the previous slide that these are just the chromosomes. Now here you look, there would be a difference between a homologous chromosome and a sister chromatid. What is that difference? First you learn what is homologous chromosomes. These are the chromosomes which are similar in shape, size and structure and basic morphology. Now one of these homologous chromosomes come from father and one comes from mother, maternal and paternal. So these two are just similar to each other. Now why do I told you about homologous chromosome during zygotene? Because during zygotene phase only these homologous chromosomes come to lie side by side. They pair up. They pair up to form synapse. They form synapse. And this complex formed by the two homologous chromosomes is referred to as a bivalent. Also, after synapsis occurs, there is also formation of a structure called as synaptonemal complex. Now, what is the significance of this structure? This structure is used to stabilize the homologous chromosome till the time of crossing over. We will learn what is crossing over, but we can just consider the synaptonemal complex is a complex of the complex structure which is formed to stabilize the homologous chromosome, because these homologous chromosomes are paired to each other, they are synapsed to each other. Okay, now these terms homologous chromosome, synapsis, synaptonemal complex and bivalent are very important for the competition purpose. They can ask you that at which stage homologous chromosomes pair up, form a synapse, at which stage synaptonemal complex is formed. So these are very important terms. So this is the representation. I have shown the two homologous chromosomes. These are the other two. These homologous chromosomes has been shown with different colors. One is shown with black and one has a simple outline of black. So these are the two homologous chromosomes and these are the other two homologous chromosomes. And they are forming a structure called as a synaptonemal complex. And their pairing is called as synapse. These are the centrioles that had already duplicated. Now they have sent away spindle fibers. Next is the Pachytene stage. Now this is the stage during which homologous chromosome, which were two in number, just two, appear as a tetrad. Tetrad means they become 4 in number. Why? Because the sister chromatids of each homologous chromosome appears during this stage. These sister chromatids were already present because of the replication of DNA during the S phase of interphase, but they appear during the Pachytene stage only. That is why we form a tetrad. Now these sister chromatids appear, and the two sister chromatids of the homologous chromosomes are referred to as a non-sister chromatid. Now I'll explain this through a diagram. Okay, so these are the two sister chromatids and these are the two sister chromatids. These two, the two which are lying towards the ends, will be forming the homologous chromosome. They were forming the homologous chromosome. Now what appears are their sister chromatids. These sister chromatids are these two which are crossing over, are called as a non-sister chromatids. Now what is crossing over? What is the basic process? Crossing over is a process in which the two non-sister chromatids come to lie in very close association with each other, Now after the process of crossing over, there is recombination that is done. What is recombination? That can be explained. Some fragment of this sister chromatid would go up into the other part, like other sister chromatids. There will be a breakdown of a part of a fragment of non-sister chromatids by the action of recombinase enzyme, and then the exchange with the other non-sister chromatid. That means the two non-sister chromatid will exchange the genetic material in between themselves, and this process involves first breakdown of a small fragment from each of the non-sister chromatid, and then exchange of this small fragment, and finally ligation of these to the newly formed structures by the help of an enzyme called as ligase. Okay, so the process of crossing over would be more clear once I'll take you further. You'll actually see that yes, crossing over has taken place. Next phase is Diplotene. Diplotene is the phase during which crossing over is complete, and there was a synaptonemal complex which was stabilizing the homologous chromosome during the process of crossing over. This synaptonemal complex would dissolve during the Diplotene stage, but except for the main point of crossing over, that is this point and this point, it will remain. Otherwise the synaptonemal complex would dissolve. This causes the two homologous chromosomes to move away from each other. Now there is another term which comes during this is chiasmata. What is chiasmata? The presence of synaptonemal complex at the point of crossing over is referred to as chiasmata. This is the point where actually the two homologous chromosomes still remain attached to each other. The two homologous chromosomes will still remain attached to each other at this point, and this is called as the chiasmata. Now this is the Diplotene stage. Now you can see this white, this color fragment has attached to the whole black sister chromatids, and this black colored small fragment has attached to the transparent sister chromatid. This means there's an exchange of genetic material that has occurred. Now during the Diplotene stage, there is a points that are formed which are called as a chiasmata, and the spindle fiber is forming, and the two centrioles are moving towards the opposite poles. Now next we come to diakinesis. I'll explain terminalization of chiasmata during diakinesis. Here the spindle assembly, as you were seeing that it was proceeding. Now here the spindle assembly is almost complete. The spindle assembly is done. Mostly it is completed. And one more thing that is important is terminalization of chiasmata. Chiasmata was formed. Now terminalization is a zipper process. Just like a zip opens, the terminalization also occurs. How? Basically the chiasmata would disappear by slipping off towards the tip of the chromosome. This is the terminalization. First it was stick over here, then it came here, then finally towards the tip of the chromosome, and finally you will be seeing that these two split from each other. So this is the main thing is terminalization of chiasmata that occurs through diakinesis, and this process is a zipper process. Just as the zip opens, you consider or imagine a zip opening. In the similar way, first it was stick over here, then it came sticked over here, then here, and finally the two are going to separate from each other. This is how the terminalization of chiasmata occurs during the diakinesis stage. So this is the whole process of prophase I, which is further divided into these sub-phases. Now further we will learn about the other phases of meiosis I in the subsequent presentations. Thanks for watching.